I just want to remind us that before we spoke about biomolecules, we spoke about those water components. So make sure that you review those water components, compartments, the ICF, the ECF, and make sure you have an understanding of that. Then we moved on to biomolecules. So we'll talk about the 4 biomolecules here just as a review. And what were those 4 biomolecules? Lipids, right? Carbs, thank you. So lipids. We had carbs or carbohydrates. Nucleic acids. And our favorites, proteins, right? Proteins. Now, which of these were polar versus which ones were non-polar? And what does polar mean? All right, so let's reword that question. Which of these play nicely with water, right? They'll get along nicely with water. They'll get along nicely in aqueous environments. Proteins, right? Yep, nucleic acids and carbohydrates, right? So these are polar. And that basically means they have an uneven charge, and that makes them play pretty nicely with water, okay? Lipids we know are not, right? They're non-polar. And all that means is that they have a pretty even, pretty stable charge around the molecule. So even, stable charge. Okay? Now, what property of lipids make them an important part of our membrane? The phospholipid bilayer, right? That amphipathic nature of the phospholipid bilayer. So we want to really remember that our membranes are an important structure that are formed by lipids, specifically phospholipids. And the amphipathic nature of phospholipids, right? Their ability to interact with aqueous environments on the one end and non-polar, non-aqueous environments on the other is really integral to that function. They're the whole reason that we can be compartmentalized, right? All right, let's move on to question 2. What is the end goal of cellular respiration and what are the 4 processes involved? And we'll briefly go over those 4 processes. So what was the big picture of cellular respiration? Right, to synthesize ATP, right? And how did we synthesize that ATP? What did we have to do before we could make ATP? How did we make that ATP? Right, good. We had to break down a substrate, one of those could be glucose, to harness that energy to then make ATP, which can be used to do cellular work, okay? So the overall goal is to harness the energy, right, from our biomolecules, which are our proteins, our fats, nucleic acids, and we spoke extensively about carbs, biomolecules, to synthesize ATP. Oops, misspelling this completely. To do cellular work. Okay, now what were the 4 processes involved? And let's just describe them briefly. The first one was glycolysis, correct. So there was glycolysis. Then the linking step, that intermediate step. Then the Krebs cycle or the TCA cycle. And then finally, and most importantly, the electron transport chain, okay? Now let's look at some features of each of these steps or each of these stages. Now, what is this location of glycolysis? Let's talk about the location. What's the location of glycolysis? Where in the cell does it take place? The cytosol, right? And what is the overall picture of glycolysis? What's happening in glycolysis? And how does that relate to this synthesizing ATP? We know we're breaking down glucose. We're taking one of our biomolecules, we're bringing it into the cell, we're going to start metabolizing it. We're starting these series of enzymatic reactions to break it down. But what is also happening as we break down that glucose molecule? What's being synthesized or released or created as a result? We synthesize pyruvate as our end product, but what's also being released? A little ATP, correct, right? So we get some ATP, 2 net to be exact. What else do we get from glycolysis? We get some NADH, right? The reduced form, which means it's shuttling some electrons. All right, so we get some NADH, okay? And we know this is going to where? Where are these electrons being shuttled? To the electron transport chain, which happens in the mitochondria, right? So those are heading to the electron transport chain. What about this linking step? What's happening in this linking step? What's the substrate and what's the end product? Right, so pyruvate is our substrate. And what's our end product? Acetyl-CoA, perfect. And that acetyl-CoA goes where? To the next step, right? To the Krebs cycle. Okay, now we get to the Krebs cycle, and what's the substrate of the Krebs cycle? We know it's acetyl-CoA. What's the overall goal of the Krebs cycle? And what are the end products that are released? So it's really important in the Krebs cycle that we create additional NADH+, but also FADH, right? And then also in the Krebs cycle, we release carbon dioxide as our metabolite, right, as a byproduct of this process. So we have NADH plus and FADH, 2 plus. We also create carbon dioxide as a byproduct. Okay? And then finally, the electron transport chain, which is probably the most important part of this. What's the most important feature of the electron transport chain in terms of the energy synthesis or ATP synthesis? No, go ahead, I'm here. Oxidative phosphorylation takes place here, right? So oxidative phosphorylation. How many ATP do we release here through the ETC? Not 43. 28 to 34, right? 28 to 34. So we make 38 net for this. For one molecule of glucose, we make 38 net. Just from the electron transport chain, we get 34 of that max, right? Some textbooks will say 28, but around 34 of that. The other 4 are coming from the previous processes. What else do we want to remind ourselves about the electron transport chain without going into excess detail? What are some of the key pearls of that process? So it synthesizes the most amount of ATP in terms of breaking down a molecule of glucose. So it's the most efficient of these 4 processes. But what else is important about this step? We said oxidative phosphorylation. What else happens here? It's taking place across the inner mitochondrial membrane, right? And those electrons that we brought in, right? Those are what are driving the process of essentially pumping those hydrogen ions, right? We talked about that chemiosmotic coupling, that creating the hydrogen pump. So we have more hydrogen ions on the outside of our membrane, the intermembrane space, and that will then drive those hydrogen ions down ATP synthase and synthesize 34 ATP for one molecule of glucose. Okay, so make sure you brush up on the ETC, make sure you understand what's taking place, where the concentration gradient is, where the pumps are, etc. All right, let's move on to question 3. What are the basic components of the fluid mosaic model? And we're going to talk about what they offer to the membrane, what feature, what function. So the basic components of the membrane. So I'll jump off and start here. We already said phospholipids when we talked about the biomolecules, so I'll write that down. And what did phospholipids offer to the membrane? Sorry? Right, so it made the bilayer, which is really important. And what about a phospholipid? We said that it was amphipathic. What else did it offer the membrane in terms of functionality? Why do we call this the fluid mosaic model? Because it's not rigid, right? It has fluidity, it has flexibility, right? Which is given because of these amphipathic phospholipids. So fluidity and flexibility. All right, what are the other structures? We had our peripheral proteins, right? And we had what other types of proteins? Integral proteins. Peripheral proteins were on what side of the membrane? Cytosolic side. Whereas integral proteins were one of two flavors. What were the two types of integral proteins? Transmembrane and one-sided, right? So we had a transmembrane versus a one-sided. What were some examples that we looked at of transmembrane proteins? What were some examples of transmembrane proteins? Right, one of the pumps, exactly. We talked about our carriers, the channels are transmembrane proteins, and our pumps are transmembrane proteins. All right, so just bear that in mind. What else is in the membrane? What other important structures are in the membrane? Cholesterol, yep. And what does cholesterol do? We said one word in terms of cholesterol's function in the membrane. So it helps to break up those fatty acid interactions. So it helps to prevent crystallization, right? So it prevents crystallization. In other words, it improves the fluidity as well. All right, what else is in the membrane? We talked about carbohydrates, which were attached to either lipids or to proteins, right? Glycoproteins or glycolipids, and those are mainly involved in cell tagging. Okay, so make sure you brush up on the components of the membrane, right? Especially those phospholipids and why they offer the membrane fluidity, flexibility, right? Their nature. Okay. Were you guys done with that first period? I'm so rude. Okay, this is probably a bit too bright, isn't it? Okay, so we'll move on to the other questions, which talk about active and passive transport. Very important that you're able to distinguish between active, passive transport, and then those subtypes that we talked about, okay? So are we done on this first one? Yep, okay. Okay, so let's move on to speak about the driving forces now, those chemical and electrical driving forces. So what driving forces act on a particle to move across the cell membrane, and what influences each of these forces? Let's start by looking at some terminology, right? What is the membrane potential? What is the equilibrium potential? What is the chemical driving force? What is the electrical driving force? And then what is the electrochemical driving force? Okay, so how do we describe the membrane potential? What is it? The membrane potential, right? When we say potential, we mean a force, right? So what is that force across the membrane? What is causing that force? It's that uneven distribution of charge, right? The unequal or uneven distribution of charge across the membrane. Oops. Okay, so remember we said that because the membrane is compartmentalized, because we have the ability to create one environment on one side of the membrane and another environment on the other, we can then have uneven distribution of that charge, right? We can have negative inside the cell, positive outside the cell. And that creates this mechanism where we can do work from that, right? Similar to a battery. That distribution, that unequal separation of the charge is a potential to do work, right? A force. What is the equilibrium potential? What is the equilibrium potential? Now, we talked about the word equilibrium a lot, right? So equilibrium means that we want to be at equal distribution across that membrane. So what's the force that wants to do that? The concentration gradient. Good. So the concentration gradient is what determines the equilibrium potential. Okay? Now, what is the membrane potential of an ion at equilibrium? What did we say happens at that membrane potential? What's going on with our two forces? So if we say an ion is at equilibrium, what is happening across the membrane with respect to that ion? When an ion is at equilibrium, the forces are equal and opposite, right? The force that wants to push it into the cell wants to push it out of the cell. And so where does it go? Nowhere, right? It doesn't move. So when an ion is at equilibrium, it's at that membrane potential where it's not trying to move, right? It's equalized across the membrane. All right? So our electrical driving force is equal and opposite to our chemical driving force, and so we have no net movement. That's what happens at equilibrium. Right? The chemical driving force. Let's talk about the chemical driving force. So what is the chemical driving force? Perfect. So the force that's moving down or along the concentration gradient. Okay, simple. So that's based upon the concentrations on either side of the membrane. What about the electrical driving force? This was based on two things. What were the two things that we mentioned? The charge on the particle and what else about the cell creates an electrical driving force? The polarity of the cell, the ability of the cell to be negative on the inside and positive on the outside. Okay, that creates this environment where we can have opposite charges attracting and like charges repelling. Okay, and then the electrochemical driving forces is simply the sum of the two forces, right? The sum of these two forces. Okay, Now let's talk about our little flow chart that we did to determine the direction of the electrochemical driving force. And we talked about 3 different scenarios. We talked about if the membrane potential was greater than the equilibrium potential. We talked about if the membrane potential was equal to the equilibrium potential, and then if the equilibrium potential was greater than the membrane potential. Right? So if our membrane potential is greater than our equilibrium potential, what's the direction of our electrochemical driving force? Perfect, right? So the electrochemical driving force will favor the electrical driving force, okay? What about if they're equal? No movement, right? This is 0, no movement. What about if the equilibrium potential is greater? This word, okay, good. So it favors the CDF, correct. So if the equilibrium potential is greater than the membrane potential, then the electrochemical driving force favors the chemical driving force, okay? And you should be able to discern that. Even if you have like a scenario, make sure that you understand what's happening here. I'm not gonna go into excess detail about this again, but go over that flow chart that we went through and make sure that you can understand where the electrochemical driving force would go, whether into the cell or out of the cell, okay? And then lastly, let's look at the types of cellular transport. We talked about 2 main types, and then we talked about some further subtypes. Okay, so what were the 2 main types of transport we spoke about? The 2 main types of cellular transport. Active and passive, right? Active or passive and active. Okay. What were the unique features of passive diffusion? Unique features of, sorry? Sorry? Oh, spontaneous, I'm sorry. Spontaneous. We also said downhill. What was another word that we can use in terms of the energy requirements of passive reactions? Are they exergonic or endergonic? Are they creating energy or are they requiring energy? They're creating energy, so they are exergonic. Okay? Whereas active transport was non-spontaneous, so just the opposite. It was uphill, which means it required energy and therefore endergonic. Okay? So let's talk about the subtypes here. We talked about 3 types of passive transport. And again, just to remind ourselves what all that these words really means is that it doesn't require energy, right? It's going purely off of the concentration across the membrane. So what were the 3 types we spoke about? Simple diffusion, and then facilitated. Yep, and then we spoke about another type, moving through channels, right? Facilitated through channels. Good. And then active transport, we talked about 2 main types. Very simply, what were those? Primary and secondary. Okay. Now let's talk about the important features of simple diffusion. What was the only feature that determined simple diffusion? The only feature that determined the rate of simple diffusion, or determined simple diffusion, not the rate. The concentration gradient, right? So, I'm sorry. So this is determined by the concentration gradient. Okay, what was an example that we talked about for simple diffusion? Oh, no, sorry, wrong one. Okay, so simple diffusion is purely according to the concentration gradient. Particles move from high concentration to low concentration as long as they're permeable to go through the cell membrane. What are some factors that determine the permeability of a solute or a substrate? Talked about factors that affected the rate and factors that affected the permeability, right? Make sure you're not confusing those two things, because you can speed up the rate, but if it's not permeable, if it's not likely to go through the membrane, you're not influencing the permeability. And you can influence the permeability, which also influences the rate. So what were the factors that affected the rate? We said the strength of the force, so the strength of the concentration gradient. If you had more of a gradient, you had a faster force, right? We also said the permeability, the likelihood of that molecule to go through the membrane. And then we also said, what was that last one we said? Influence the rate. The surface area of the membrane. How much surface area is exposed is going to determine how fast we have movement through that membrane. All right, so make sure you brush up on simple diffusion and you understand the factors that affect the rate and the factors that affect the permeability of the membrane and of that process. Facilitated diffusion. What did we say about facilitated diffusion? So this required a protein. What type of protein is it an example of? We talked about this earlier. So this requires transmembrane proteins. Right, transmembrane proteins. And what was an example that we talked about with facilitated diffusion? Sorry? Not the sodium potassium pump, that was active. What was an example of facilitated diffusion? We looked at the uptake of a specific substrate, glucose, right? That GLUT4 transporter. It sits in the membrane, it interacts with glucose on the outside of the cell. There's a configurational change in that protein. Glucose is exposed to the inside of the cell, and so it comes to that transporter or that carrier, right? So our example was the GLUT4 transporter. Okay, the important feature that distinguishes facilitated diffusion specifically with carriers is that the substrate or the molecule interacts with one side of that protein at a time, right? It doesn't go through it the way it does for a channel. All right, so for a channel, this is also a transmembrane protein. And what were some examples that we spoke about for channels? What were some examples that we spoke about? What are some things that actually need to go through a channel because they cannot go through that lipid bilayer? We spoke about aquaporins, correct? Aquaporins. And aqua, can't, okay. And what molecule do aquaporins facilitate or allow? Water, good. What are some other channels? Now we know charged particles can't go through the membrane, so they need a channel, right? We also spoke about ion channels. Okay? All right, so the take-home point about channels is that they can interact with the membrane on both sides, right? There are also bidirectional channels which facilitate the bidirectional movement of ions, and then they can also facilitate the movement of water, so aquaporins. All right, let's look at active transport. So we spoke about two types, primary and secondary, and what was primary? And what was an example? So we know this requires energy. One of them is going to require the synthesis and the metabolism of ATP. And which one was that? Which of these processes required ATP? Primary, thank you, right? Primary. So it requires hydrolysis of ATP. Okay, what was an example that we spoke about? What was an example of primary active transport? So we spoke about a really important structure in our cell membranes at the basolateral, the sodium-potassium pump. Perfect. So the sodium potassium pump. Okay. And then finally, we spoke about secondary transport. And what was the feature of secondary active transport? Perfect, right? So the energy was released from another ion movement. All right, in this case, we spoke about an example where what was the ion that was moving? What was the example of secondary transport we spoke about? Anyone remember that example? We spoke to the sodium-linked glucose pump, right? The sodium-linked glucose pump. And that was basically an example of what type of transport? Well, it's secondary and it's active, but we called it another name. Because both of these ions are moving in the same direction, we called it co-transport. And then we looked at another example where the ion and the substrate in question are moving in opposite directions. And what would we call that one? Countertransport. Good. Make sure you brush up on that terminology. You will definitely be seeing that on the exam. Okay. As far as review, oh, I'm so sorry again. Okay. As far as review topics, I think that's all I'm going to cover. I'm going to address some questions that I got via email quickly, and then I'll see if there are any other questions here. And we will okay, so before I go into this, I got questions on positive and negative feedback, so I'll just brush up on that. The depth of cellular respiration. So as far as what we went over for cellular respiration, that's probably the most important components, the parts that you really need to focus on. Don't worry about the enzymatic steps. Don't worry about memorizing the names of any of those intermediates. You really want to know the substrate, what goes into that reaction, the end product, what comes out. Okay, that's what you really want to focus on, and not so much that the fine details. And then I'll also talk a little bit about active and passive. Well, I think I just reviewed active and passive transport, but if there are additional questions, I'll take those at the end. And while I'm pulling this up, guys, I really want to encourage you all to practice questions. I think that's probably the most important part of staying prepared and mastering the material. There are questions in your book. I typically bring questions to lecture. You really want to practice recalling that information. Okay, that's how you're gonna master it and get good at it. Practice, you know, as you're sitting, just try to remember it, right? Research shows that just trying to remember something is how you really ingrain it in your memory and are able to master it so that you can recall it when you're taking the test, right? Okay, so I'm gonna go over positive and negative feedback first. Just very briefly, so the pearls of positive and negative feedback. Okay, so we spoke about homeostasis and we spoke about this loop of homeostasis. And using this as an example, we said that the body has this mechanism to potentiate or to activate certain processes if it were needed, and then this mechanism to shut those processes down if they are no longer needed. All right, now looking at this example of blood pressure, we have a stimulus, which is standing up quickly, right? Standing up too quickly, whether we realize it or not, pulls most of our circulation to our legs. And so without our conscious proprioception, our body determines hypoxia, right? There's some areas of the body that are not getting as perfused as others. That sets off a stimulus, which is picked up by a sensor, right? We have tons of sensors, baroreceptors, chemoreceptors on our blood vessels, and they can determine whether there's enough oxygenation coming in or not. That message is then sent to the integrating centers in the brain, and then from the brain, the medulla decides, look, I need to increase the blood pressure. So that is the effector signal. And that is sent out to the organ that will most likely be able to increase the blood pressure, which is the heart, okay? So we send this signal down to the heart. We'll talk about the exact fibers because this is a parasympathetic signal here. And then as we increase the blood pressure, those receptors are those sensors are no longer picking up that stimulus because we've now evened out our perfusion, and those areas of our body that sensed hypoxia when we stood up are no longer sensing that hypoxia. And so what we need to do at this point is turn that signal off. All right, so closing this loop is what negative feedback is. Going back to the original sensor and saying, look, I've corrected the error signal. You can turn off that sensor, you can turn off that signal, you can turn off that initial relay. All right, that's what negative feedback is. That's why we call it a loop, because it's the closing component of making this a cycle. All right, that negative feedback loop. Let's look at this image. So this is the same sort of image here. We have a stimulus, goes to a sensor, our integrating centers, then the effect comes along, but in order to close this loop, we have to inhibit or negate that initial stimulus. Otherwise, it will potentiate forever and ever and ever, and our blood pressure would rise indefinitely, okay? And that's not what we want. That will be working against homeostasis. So closing that loop, turning off that signal is an important part of maintaining homeostasis. Let's look at positive feedback. Now, positive feedback is opposite of this scenario. So this is where, for a brief moment, again, not indefinitely, because that would negate homeostasis, for a brief period in time, we want to potentiate that signal. We want to amplify it. We want to make it more impactful, right? So an example here was the secretion of luteinizing hormone from the pituitary gland. And that would then potentiate the ovaries to secrete estrogen. Now, there's a time along the menstrual cycle where we need to have a surge of LH in order to stimulate ovulation. So this is a part of the mechanism in terms of our reproductive function that requires a positive feedback loop, okay? And so what happens here is that this estrogen secretion further potentiates more LH secretion. And this goes on indefinitely until we have a spike in LH and a spike in estrogen such that we can stimulate that ovary to ovulate or to release that follicle, okay? And so we don't see positive feedback loops often throughout the body, and this is why. They're not necessarily working with homeostasis because they're potentiating something indefinitely. But there are a few instances in the, or areas in the body where we do need to have a positive feedback loop. We spoke about this, which is associated with the monthly cycle. What are some other examples we talked about for positive feedback? We spoke about two other examples of contractions during birth, right? Those uterine contractions during birth. We synthesize hormones to make those contractions stronger. And as that fetus is moving through the birth canal, that cycle is potentiated, it's amplified until that fetus is born, and then you can shut that down with the negative feedback. What was another example that we talked about of positive feedback? Blood clotting, exactly. So we have an injury to a blood vessel, we sequester clotting factors, they lay down at the site of injury. We don't want that to stop, like we want that to keep going for a while, right? So those clotting factors keep coming, laying down on and on because of a positive feedback loop. When that injury is healed, that will then become a negative feedback loop because we no longer need it, right? And that is how we can